In this part, we are going to look at the senses of equilibrium, of taste, and of smell. But let's review what we looked at for the eye and for the ear. So the eye has three tunics. A fibrous tunic made up of the sclera and cornea, a vascular tunic made up of blood vessels, the colorful iris, the lens, the ciliary body, and the choroid coat, which prevents light from scattering. And then the neural, or the internal, layer is the retina, which has photoreceptors. If we were to look on the periphery here, we would have rods that converge onto single bipolar cells. So two rods converge onto one bipolar cell, and the bipolar cells converge on a ganglion cell. If you were to look at the macula, which is right here behind the lens, you'd have lots of cones, and each cone has its own bipolar cell, and each bipolar cell has its own ganglia cell. So light comes in, gets refracted by the cornea, gets refracted by the lens, and it hits the photoreceptors, either cones or rods or a combination of the two. And the signal goes to the bipolar cells, which go to the ganglia cells, which then go out the optic nerve. Now right here, where the optic nerve leaves, we have the blind spot. Now for sound, we have sound coming in the external acoustic canal and vibrating the three ossicles. I do not know what happened to my malleus right there, but that pushes on the incus, which pushes on the stapes, which pushes on the oval window. The oval window is attached to this snail-like structure called the cochlea, and it's actually two tubes. You have this big continuous tube forming a second tube in the middle of it. Now this middle tube is the cochlear duct, and that's where these hair cells, which are the organs of hearing, are actually located, or the receptors of hearing. Now, the top border is called the vestibular membrane, and the bottom border is the basilar membrane. The vestibular membrane forms the bottom of the scale of vestibuli, and when sound comes, the waves are going to go through and move the perilymph. If it's a high-pitched sound, like Ariana Grande, it's going to hit here and cause an action potential or signal from these hair cells. If it is a deep bass voice, it's all about the bass, you know. The sound waves are going to go farther, hit it closer to the apex, and excite the hair cells that are there. And whatever combination there is of these hair cells being signaled, you hear as music or your teacher talking or your cat meowing or whatever you happen to be hearing. So now we're going to move on to equilibrium. So we have the receptors of sound right here in the cochlea, and the oval window is actually there in the vestibule. Now there's receptors for equilibrium in both the vestibule, which is this structure right here, and the semicircular ducts, which are here. And we're not going to worry about the anterior, lateral, or posterior ducts. What we care about is these are tubes, and there's tube-like structure in there, and they have endolymph. And when you are moving your head, the endolymph moves through the semicircular ducts. When you're stationary, it should not be moving in the endolymph in the vestibule. And this gives us two types of equilibrium: static equilibrium and dynamic equilibrium. Static equilibrium is equilibrium of standing still. It's telling you your head is stationary and you're not nodding off listening to part 25 of a lecture on the special senses. This is only part 4, by the way. Dynamic equilibrium is telling you your head is spinning, and it's trying to make sense of that spinning. Static equilibrium uses hair cells in the vestibule. And let's say you're falling asleep in class like this person is. Oh no! That's going to shift the endolymph in the head, in the vestibule, and it's going to trigger the hair cells. And the hair cells are going to be like, oh, we're falling asleep in class. Oh no! Let us wake up. So it lets you know your head just moved. So static equilibrium is telling you about the stationary position of your head. 3 underlines, stationary. Now, dynamic equilibrium reports on movement, how your head is turning. And the endolymph moves through these semicircular ducts in the semicircular canals. And hopefully the left one and the right one are agreeing with each other. In one type of vertigo, you can actually have a block in one of them, and your two semicircular duct systems are not agreeing with each other. That is what gives the effect of vertigo. Another type of vertigo, you're actually pinching the vestibular nerve. So this is telling about the movement of the head. So I'm going to ask you one of the two types of equilibrium. I've even gotten desperate to the point where there's answer A and answer B. Static equilibrium is telling the stationary position of your head. Dynamic is reporting on movements of your head. Still half the class gets it wrong when I ask. We ran out of time on our ear lecture. So real quickly, tell me the functions of these because I'm going to ask you them on the test. Auditory tube is actually another name for the eustachian tube. It allows for equalization of pressure. I hate the word auditory tube. It has nothing to do with sound. I do not know why they changed it. The auditory canal takes sound to the tympanic membrane. The cochlea has hair cells, which are receptors for hearing. The ossicles amplify sound. The auricle funnels sound into the auditory canal. And the tympanic membrane basically creates mechanical energy from the sound, so it vibrates. Which one contains hair cells? Well, I gave it away on the last slide. It's the cochlea. So we're going to look at senses of smell and taste. They're both chemical senses. And we're going to look at the papilla. I've never been really big on the different types of papilla. We're going to identify the top parts of a taste bud and see how smell and taste are related and look at the nasal epithelial. So gustation is taste. Olfaction is smell. And they're using chemoreceptors to detect chemicals. So you have these olfactory cells up here in your nasal epithelial. They are going to detect chemicals and send the information on cranial nerve 1. You have taste buds in your tongue and actually on your pharynx. They are going to detect chemicals in what you eat, and it's going to send signals on either the facial or glossopharyngeal nerves. Now notice, the two cavities are connected. You could actually put a piece of spaghetti in your nose, grab it in the back of your mouth, and use it as nose floss. I don't know why you do that, but you can. So things that you eat, you're also smelling. So taste is related to smell. So olfaction is the fancy way to say smell. Now here is cranial nerve number 1, the olfactory nerve. And there are these bipolar cells sticking down. And what happens is you smell something, let's say vanilla, and that binds to a certain combination of receptors. Then you smell skunk. That binds to a different combination of receptors. And it takes the signal on the olfactory tract to the olfactory cortex, to the amygdala, to the temporal lobe, probably to every single possible lobe because smell is really complicated. Now, here's the bipolar cells going through, and they're calling it cilia attached to it. They're really just the extension of the dendrites. That's it for smell. So taste. Taste uses gustatory cells that are formed in taste buds. In second grade, you probably learned you have one taste at the tip here, one taste on the side. The teacher lied to you. That's not true. Each set of gustatory cells actually has all 4 or 5 or 6 different types of taste buds. And that study that says you could taste each one at different places, it had a very bad protocol that we repeat to this day So when your first grade teacher does that, you get the same results. So, taste buds are on these structures called papilla. These are my favorite papilla right here. They're the valate papillas. And they're on different parts of the papilla. So you have some valate papillas here. You'll have fungiform papillas. So the taste buds are located. So here's the valate papilla, and the taste buds are here on the side. So what has to happen to whatever you just ate, that cheeseburger, is the little parts have to dissolve and then get into this little valley in the valate papilla. More about the taste bud, though, than the papilla. So we have these gustatory cells. This one might be for salty. This one might be sweet. One might be for sour. And this one, my least favorite, is bitter. Now, they're in little microvilli that stick out. Remember, microvilli extend surface area. And chemicals bind to them. If it is something that's bitter, it will activate this taste bud. If it is something sweet, it's usually a ring structure, it'll activate the sweet taste bud. Sodium and potassium will activate the salty taste bud. And then hydrogen ions actually activate the sour ones. And that's how you taste it. So you have these little taste buds. The chemicals get in, go through here, and it activates. And it sends signals to the brain. There are 3 good tastes. Salty's telling you there's sodium and potassium. It actually goes to your pleasure centers, telling you the food is good to eat. So you're going to want to eat more and more. That's one reason they put salt in a lot of food. Sweet. Fructose and sucrose are a ring-like structure. They activate your sweet taste buds. That also goes to your pleasure center in your limbic system. So when I was a kid, they took all fat out of food, and they had to replace the flavor with something, so they put high fructose corn syrup. So a lot of manufactured food is high in salt, so you eat it, and high in sugar, so it tastes good. This activates your pleasure centers, and you want to eat more. Umami means savory in Japanese. That is telling mainly glutamate, which is the main excitatory neurotransmitter, but it detects other amino acids as well. Now, all 3 of these are rare in nature. Good sources of sodium, good sources of sugar, good sources of glutamate. So all of them go to your limbic system. So if only I could come up with like a McCrack burger that has all 3 of those substances. Oh, wait, McDonald's already did that. There's 2 bad tastes, and notice I put bad in parentheses. Our bodies evolved, yes, I just used the E word, to say these are going to be bad things in food, so we shouldn't eat the food. Bitter is telling you about alkaline toxins. So if you ate soap, it would be bitter because soap is basic. Now, a lot of plant poisons are bitter. It's telling the herbivore, don't eat me, so they'll spit it out. I once got a cane toad, they're really cute, shoot its poison right next to my mouth. It was probably an inch away. It's such a strong toxin, I could taste it inside my mouth, even though it did not get inside my mouth. It's telling you the food is bad. So, that is bitter. Did you know coffee and chocolate are both bitter in real life? Those are plant toxins. I love both of them very much, by the way. Sour is telling about hydrogen ions. Either the food is unripe or it's gone bad. I have 2 lemon trees in my backyard. I love sour food. So we've decided, oh, sour actually isn't that bad. We'll eat sour food. Other tastes. Spicy is not a taste. Underlined right here. It is pain receptors from peppers. Those are free nerve endings, remember that? So this guy eating a pepper, he's actually going to stimulate pain receptors, those free nerve endings. Metallic ones are debated. There's one side of the story that I'm on, that you have other taste buds are telling you about these divalent cations like copper, iron, magnesium, even calcium. Others saying it's actually stimulating both the bitter and the salty. I could actually see that side of the argument too. So that's why I put it as a question mark. Lipids are debated too. According to my nutrition books, which are now 15 years old, It could just be the texture of the lipids that goes to your pleasure center. Others claim it's receptive.